Okay, so our next speaker is Vivian Pera, who's working with Mark, Nidra, and me.
That's interesting.
Great, I'm glad you think so.
Not so you're working on
think so?
non-negative too.
So, I'm not quite sure why mine gets stretched out here, but I don't think that's going to impact what I have to say too much. I couldn't figure that out earlier. But anyway, as Dana said, I'm Vivian Pera, and I want to thank the NEPSSS committee for inviting me to speak, and all of you for attending. So my research is really at the intersection of biomedical optics and signal processing. And today, what I want to talk to you about is the demixing problem. So what do I mean by that? Well, suppose you have a signal, a measured signal, which is what I'm indicating here with the black trace. But you know that this signal is a mixture of some other signals. In this case, it's the sum of the 4 signals which are displayed here in color. The demixing problem is essentially how do you recover the underlying signals from your measured signal? Now, for me, this problem arises in the context of fluorescence molecular tomography, FMT, in particular something called multiplexed FMT. But as you can see, this description is quite general, so this problem arises in a number of other fields. And likewise, the algorithm, the demixing algorithm that I'm going to share with you today is also quite general, and so therefore could be applicable in areas outside of FMT, perhaps even to the research of someone sitting in this room. So what I'm going to do today is I'm going to start out by giving you an introduction to multiplexed FMT, and then I'm going to go ahead and present the algorithm, and we're going to look at its performance under some ideal and not so ideal conditions. And then I'm just going to wrap up with some conclusions. Okay, so what is fluorescence molecular tomography? Well, it's an optical technique that uses near-infrared light to map the 3-dimensional distribution of fluorophores in a whole animal non-invasively. In our lab, we work with small animals. We work with mice, so that's why we have a picture of a mouse here. And if we want to talk about FMT, basically the first thing I need to do is talk about a fluorophore. So for the purposes of this presentation, I'm going to define a fluorophore as a chemical compound that absorbs light at shorter wavelengths and then re-emits it at longer wavelengths. And the important thing to know about this is that fluorophores have characteristic emission spectra and lifetimes. So in wavelength, they usually have a peak emission, and then it sort of, decreases in this kind of bell-shaped pattern. And then in time, the signal follows this mono-exponential decay model. So the lifetime parameter that's used to characterize a fluorophore is just simply how long does it take to go from the peak down to the 1/E point. Now, those of you who work, whose work connects somehow to the life sciences may know that, in recent years there's just been an explosion in the number and variety of fluorescent probes that can provide contrast against specific biomolecular targets. So doing this kind of, doing fluorescence imaging is incredibly useful. It can give us both structural and functional information. For example, you can use fluorescent dyes to stain particular components of cells and look at really high contrast images. You can also genetically modify cells to express fluorescent proteins, and so therefore you can see when and where certain genes are being expressed. So, this could be a whole lecture in and of itself. So just suffice it to say that this is a very useful thing to do, and the goal of FMT is to be able to generate a 3-dimensional image in a whole animal non-invasively. So how do we do that? I'm sorry?
It's injected into the mouse?
The fluorophore, it can be injected. Other times you have, for example, you can transfect cells in a tumor and then implant it. So, but it's somehow introduced
Inside
into
the mouse.
Yeah, it's inside the mouse, and we want to know where it is without cutting into the mouse. So the way we do this is similar to the way X-ray computed tomography is done. So basically, we surround the mouse with a series of sources and detectors. And I have to say, this is a notional image. So if we wanted to image around the torso of the mouse, we would do it around the torso of the mouse. And also the fluorophore is not located in a little, compact spot either. This represents the location anywhere in the mouse. So basically what we do then is we illuminate the animal with a laser, and we do this all around the animal to come up with this dense web of source detector pair measurements. So now, unlike X-rays though, near-infrared light in tissue propagates according to a different model. It basically does this kind of directed random walk. And so if I map where light that gets introduced at this point and detected at this point, if I map where it goes in the tissue, you come up with these kinds of banana-shaped volumes. And what this means for your image reconstruction problem is that your resolution is limited. And the image reconstruction problem becomes what we call an ill-posed inverse problem. So typically the next step, the way that we generate the image is we form a system of equations where on the left-hand side we have our measurements, our various source detector measurements all in one long vector. Then we have this weight matrix which incorporates the model of light propagation. And then we have this vector which is the image, a vectorized image that we're trying to recover. And because this problem is ill-posed, in order to be able to invert this matrix, we need to regularize it. And what that means is that the image that we generate will depend on the assumptions that we use to regularize the problem. But, this is the physics of the propagation. We have to do this, and so people do it, and you can come up with an image of where the fluorophore is in the animal. And if you do this, along various heights, you can stitch that all together and come up with a three-dimensional volume. Okay, so I was saying a little while ago that fluorescence imaging is very useful in the life sciences. In microscopy, it's pretty much ubiquitous. So images like this are, very common for them. Here what we have are two cells, and we've used 3 fluorescent proteins to stain the various structures in the cell. So this is in microscopy, but when we image through several centimeters of tissue in an animal, we typically only use 1 or at most 2 fluorophores. And so the goal of this project is really to be able to image multiple 4 or more fluorophores simultaneously in whole animals. So what are the challenges? Well, the issue is that since we have to go through several centimeters of tissue, we're limited to where we can operate in terms of the spectrum here. And so what this plot shows you is the absorption coefficient of some of the typical constituents of tissue, like melanin, and we have water, deoxy and oxyhemoglobin. And you can see that they reach minimum in this area from between 650 to 1.3 microns. So that's the only place where we can get enough signal in and out in order to be able to see anything. And in fact, we're even more limited in the IR by the sensitivity and efficiency of the detectors that we use. So really what we have to work with is about 200 nanometers from between 650 to 850. And so here I've plotted the emission spectra of some common fluorophores, and what you can see is that they're rather broad. And if you want to put 4 or 5 of them in there, they're going to be highly overlapped. And so trying to demix this signal from this kind of information is going to be really challenging. And in fact, it's even more complicated than that, because the optical properties of tissue vary with wavelength. And so that means that the spectra that you would measure from the fluorophore depends on where exactly it is in the animal. But you don't know where it is, so you can't exactly predict the kind of distortion that will happen to your measurement. Often these things get shifted toward longer wavelengths, so this is known as redshifting. So the solution that we're exploring in our group is basically, instead of just using spectral data, we want to use both spectral and temporal data to see if we can get a better answer for this problem. And so what that means is that we're going to collect data in time at a number of different wavelength bins. And we're actually building an instrument to do this in our lab. Now, in the interest of time, I'm not going to go into the instrument because I want to focus on the data processing algorithm, which was my contribution to this project. Because the thing is that this multi-dimensional data set is really large. It's a computational challenge. So to give you an idea, we typically employ about 650 source detector measurements to reconstruct a 2D image. And given the spectral and temporal resolution of our system, each of those measurements is going to have 10,000 components. So that matrix that I showed you before, W, that would have about 6.5 million rows. So we're not just going to be able to invert this. It's not one of these things that's easy to parallelize or anything like that. So we're going to have to do something different. And the solution that I came up with was basically to split the problem into two parts. In the first stage, you take your mixed measurement signal and you demix it. And in your second stage, you take each of those demixed measurement sets and you do a tomographic reconstruction. And then if you want, you can put all of those together to come up with a multicolor image. So the thing is that, as you'll see in a minute, this stage is not computationally taxing at all because we operate only on one source detector measurement at a time. And then once you've done the demixing, you don't need those 10,000 components anymore. You can significantly shrink the dimension of that vector. And then this becomes a problem that has been done already in the field, and people have a lot of different algorithms and ideas for doing stage 2, including our group. So basically what I'm going to focus on for the rest of the talk is stage 1, because that's where the novel contribution is. Yes?
The matrix that you're talking about, how well or ill conditioning, can you tell us a little bit? Is it possible to identify how well conditioned is or ill conditioned situation so that
for the tomographic reconstruction problem, it's generally always ill conditioned.
Ill conditioned, okay.
Yeah.
But can you somehow distinguish or decompose that into well-conditioned part?
Yeah, I don't
It's
think
not
so.
possible.
I've never seen it done.
It's not possible.
Not in a useful way. The spectrum decays gradually.
Yeah.
So when you calculate the singular value,
Yeah, the spectrum decays gradually.
gradually,
Yeah.
I see. So it's not.
Unfortunately, there's not like a drop off and you can just throw stuff
I
away.
see. That's what I wanted to know.
Yeah.
No. Okay, thank you.
So anyway, but before I leave this, I want to make this point that, of course, the whole point of dividing it up into two stages was to significantly reduce the computational complexity and make it tractable. But there's another benefit. So this is always ill-posed, but this doesn't have to be, or is ill-posed in a less damaging way, if you will. And so if we do this first, we avoid introducing any bias into this demixing operation that we have to incur in order to do the tomographic reconstruction. Because when you regularize, what you're essentially doing is introducing bias into the problem. So this way of doing things has this nice advantage too. Okay, so let's take a look at the first stage a little bit more closely. So the first thing we do is, as I said, we're going to operate on a single source detector measurement at a time, and we're just going to loop through until we've demixed all 650 or however many we do. So we take the kth measurement, which is represented by this. It's a multi-dimensional measurement, both in wavelength and time, and we stack it into one long vector. Then we employ a linear mixing model. So what that means is that I'm going to say that my measurement can be expressed as the product of a matrix S, which I'm calling the library, and contains the potential, the known fluorophore signals that may be in the sample, and this unknown vector of combining coefficients A, and then we have a noise term. And now here I've kind of drawn it out, and I'm going to say a little bit more about the library in the next chart, but the only point that I want to make is that the library can be divided into sections, one corresponding to each fluorophore. And my combining coefficient vector can also be divided into those corresponding sections. And so what I do next is I solve for this unknown vector A sub k using non-negative least squares, in particular the algorithm of Lawson and Hansen, which is actually what's implemented in MATLAB when you do LSQ non-neg. And then once I have that vector A sub k, I recover the underlying signatures by multiplying the corresponding sections of that vector with the matrix. And that's basically it. And then we just loop through until we've demixed all of our measurements, and then we can use that as the input to stage 2. Okay, so let me say a little bit more about the library. So the goal of the library is basically to include in there a sufficient number of fluorophore signatures so that we capture the expected variability of the measured signal. Now, a naive way of constructing a library is basically to say, hey, if I have 4 fluorophores, let's say, I know what each of them looks like, so I have one signature, one column in there per fluorophore. But as I mentioned before, this isn't a very good thing to do with tomography, especially when you're imaging through bulk tissue, because the shape of that signature varies depending on where the fluorophore is. So you can't really say the fluorophore is going to look like this when you don't know where it is. So we can make a simplification. We can assume that the optical properties are constant with wavelength. And when we make that simplification, what we see is that the shape of the signature depends on the total path length. And by total path length, I mean if the fluorophore is located here, the total path length is the sum of the distance to the source and the detector. So all fluorophores located along this line, the same fluorophore, have the same signature. The ones located along this line have a slightly different signature, and this, etc. And I'm showing you an example here. And you can see that it's not a very huge difference, but actually including this variability in the library gives us a better demixing performance. So I'm going to call this the extended library 1. And in this library, we have 3 signatures per fluorophore, one corresponding to each of these different path lengths. Now, this is a simplification. In reality, the optical properties depend on wavelength. And when we factor that in, that leads to this distortion that I mentioned earlier called redshifting. And in this case, the shape of the total signature no longer depends only on path length. So here I'm showing you, this is one fluorophore, it's the same fluorophore, 25 millimeter path length. But the difference is that, you know, the blue represents the fluorophore being close to the source. The green, it's kind of halfway between the source and the detector, and the red is close to the detector. And you can see that not only are there shifts, but, you know, the peaks in the spectral channels are very different. So this is very different shapes for the same exact fluorophore. So in order to account for this, we create a library that has many more fluorophore signatures per fluorophore. And I'm going to skip over those details. Essentially, we create what the signatures would look like if the fluorophore were at every single pixel, and then we use an algorithm to prune the library. But I'm happy to talk to anyone that wants more details. But the upshot is that this extended library 2 has 344 signatures per fluorophore. Okay, so great. So now I can account for my variability. So fantastic, right? Except that something has happened. And the problem is that in going from the naive library and extended library 1, those correspond to full rank matrices. This library, which has, you know, a number of fluorophore signatures that don't vary that much from one another, is rank deficient. And so now you may be thinking, well, that's going to be a problem for that algorithm. How are you going to solve it without regularizing it? But I'm going to show you that, in fact, I can use the algorithm exactly as I've described it to you. And I'm going to explain why in a minute, but first I want to show you an example of how well this works. So we did some simulations to characterize performance, and I'm not going to go through the details. I just want you to focus on this picture here. So I'm assuming that I have this 25 millimeter circular sample, which would correspond maybe to the dimension of the torso of a mouse.
And on your screen, it's a circle.
Well, yeah,
Yeah.
on my screen it's a circle.
Yeah, that's what I'm saying. I'm just trying to tell everybody, it should be a circle, and that elliptical shape is because of the distortion of the detector.
Elliptical, okay,
Yeah.
that includes circle. So anyway, yeah. We have the source, and then we have a series of 9 detectors on the other side, and we're going to rotate that entire configuration and take measurements so that we wind up with that dense set of source detector pair measurements that I talked to you about. And then in there, I'm assuming that there are these 4 different fluorophores present at this location. And then we're going to consider 2 cases, the simple case where the optical properties don't vary with wavelength, And in the more complicated case where we do allow that wavelength dependent variation, and this is what that looks like. This is the absorption coefficient and the reduced scattering coefficient, and this is what the variation looks like across our spectral channels. And then what I do is I generate 10 realizations of each measurement set. Remember, each measurement set has 650 measurements. And I compute, I add noise, a realistic level of noise, and then I compute this error metric. And what this error metric is, this n hat contains the demixed fluorophore signatures for each fluorophore, n for the F fluorophore, n contains the true values. This is the Frobenius norm, so this is an RMS difference, and then we're just normalizing by the magnitude of the true values. So it's a normalized RMS error that we're going to use to quantify demixing.
Sorry, I have a question here. I feel
Yes.
like there should be 4 plots, so I'm missing something. These 2 plots, plot 2.
These are the optical properties of tissue.
Okay.
So this is the absorption coefficient and the reduced scattering coefficient.
Okay, so then I see 2 bullets, and one says no redshifting and one says with redshifting, so I'm having a hard time mapping those 2
Oh,
bullets.
sorry, so these 2 plots correspond to the second bullet.
Ah, okay,
So with no redshifting, this would just be a line.
okay.
That's it. Okay, so let's look at some results. So these are the results when there's no redshifting. In other words, we assume that the optical properties don't vary with wavelength. And the red squares represent the demixing performance when we use the naive library. And the blue circles represent the demixing performance when we use extended library 1, which was the one that had 3 fluorophore signatures per fluorophore. And we can see that, and by the way, the standard error are not on here because they would be smaller than the symbol sizes. So these are significant differences. And we can see that there's a performance improvement in using extended library 1 of about 6 to 10x, depending on which particular fluorophore you're looking at. Now, this was what we saw when we had the fluorophores in these positions, okay? We also were wondering, well, what would be the impact of, having them in different locations? How much would that change the results? And so what I did to assess that was, I did 50 trials where I just dropped them in randomly, and then this blue, or gray, I guess, bar here represents the minimum and maximum errors that I saw over all 4 fluorophores. So not a whole lot of variation depending on position, significant difference depending on whether you have those extra signatures in the library or not. Of course, the more interesting case is what happens when the optical properties are allowed to vary with wavelength. And in this case, you can see that if you just use that naive library, your algorithm is really not going to work at all. This is a logarithmic scale now, so your errors are up around 70 to 100%. Whereas if you use this extended library 2 with the 344 signatures per fluorophore, you can get down to between 2 to 3%. So big improvement. Under those conditions, you really do need to use something like the extended library in order to be able to successfully demix your signals. So now, I've just shown you that, it's important to account for that signal variability when the optical properties vary with wavelength. But of course, how the optical properties vary with wavelength is also not known ahead of time, exactly. And so we did some investigations to see how sensitive this algorithm was to model mismatch. In other words, if I think that there's a certain variation in optical properties, like this, for example, and I use that to construct my library, but instead the variation in optical properties looks more like this, and I use these to construct my signals, how well would my algorithm perform? And so I went ahead and did that and computed the same error metric, and this is the result for that. Now these labels correspond to numbers that tell you by how much the library is either overestimating or underestimating that variation. And one corresponds to the case where the properties between the signal and the library match exactly. And what you can see is that there's an asymmetry in the results. So it seems like it's much better to overestimate the variation than to underestimate it. And if you think about it, that kind of makes sense because, you know, if you have more variation than what you're actually seeing in the library, you at least have a shot of accounting for what you're actually seeing, whereas the reverse is not true. The other thing to note though, and I'm not showing you any tomographic reconstructions just due to time constraints, but when you actually do tomographic reconstructions with these demixed signals, you see that whenever you have about less than a 10% error, you actually get quite good tomographic reconstructions. So it does seem that we can tolerate, you know, at least from this initial investigation, some degree of mismatch in the algorithm and still get good enough results so that our tomographic reconstruction algorithm works really well. Okay, so now let me return to this question of why does the demixing algorithm work even when S is rank deficient? So here's our algorithm right here. This was our data model. We're solving this with non-negative least squares. We don't have a regularization parameter. And then we recover each of the fluorophore signatures, remember, by multiplying the section of the coefficient vector with the corresponding section of the matrix. So the first thing to note is the following. So usually when people solve this kind of problem, they're really interested in the results of that coefficient vector. You know, what is AK? That's the goal. But for us, we're only interested in it sort of in an indirect way, because as soon as we get it, I'm going to use it to reconstruct the underlying fluorophore signature, and then I'm essentially going to throw it away. So the only thing that I need, really, for us, the only thing that we need is to have that solution limited to the correct section of the vector, so that then I can reconstruct it using this third step. I don't need a unique solution for AK. So, what kind of an algorithm can give me that? Well, from the field of sparse subspace clustering, there is some work that shows that if you have a matrix that represents a union of subspaces model, and you ask for a sparse solution, you get a solution that's limited to the correct subspace. So for our problem, we can interpret our library as representing a union of subspaces. Each subspace corresponds to a fluorophore. And then if we ask for a sparse solution and combine it with this non-negativity, we see that we also get a solution that's limited to the correct section approximately. And I say approximately because there are small non-zero coefficient vectors in other subspaces, but they're really, really tiny. And as you can see, the results that we get are really good. So it works within a couple of % error for us. So, okay, great. So I need non-negativity and I need sparsity. And I said I'm using Lawson and Hansen non-negative least squares to solve this. So obviously I'm using non-negativity, but where's the sparsity? And the answer is that it's also coming from the Lawson and Hansen non-negative least squares algorithm. Because if you take a look at this code, you realize that this can be interpreted as a non-negative orthogonal matching pursuit algorithm. And those of you that are familiar with the compressed sensing literature will recognize orthogonal matching pursuit as an algorithm that gives you sparse solutions. And I mean, this is kind of interesting because, so this algorithm has been the gold standard for solving non-negative least squares problem. It's been around since the 70s. These orthogonal matching pursuit algorithms only started to come around, I think, in the 90s. So it's kind of interesting that this is a version of something that postdates it. So the way to interpret the Lawson and Hansen non-negative least squares, in my opinion, is really as a multi-purpose tool. If you feed it an overdetermined matrix, it will find a non-negative least square solution, just like people commonly use it all the time. But for an underdetermined problem that has a sparse solution, you can also use it to find a sparse non-negative solution. And the beauty of it is that this algorithm doesn't require any kind of regularization parameter. And so that's why I say that the demixing algorithm has intrinsic regularization, because it doesn't depend on any kind of user-defined, empirically derived regularization parameter. Okay, so let me just wrap up now. So in this talk, I think what I've shown you is that when you have spectral and temporal data together, you can use that to do accurate demixing of multiple concurrent fluorophores. We saw that one way of attacking this problem, because it's a huge data processing challenge, is to separate the demixing and the tomographic reconstruction parts. I've shown you that this demixing algorithm is accurate. Oh, I guess I forgot to say that it takes me about 5 minutes to demix 650 measurements on this laptop, so it's not at all computationally taxing. It's quick, it's flexible, you don't need a regularization parameter, even when the matrix is rank efficient. A key component of it is this concept of the extended library that allows us to account for the potential signal variability. And finally, our initial investigations tell us that correctly estimating the average background properties of the tissue and the general shape of that variation over the spectral channels may be enough to successfully demix the signals. And so with that, I want to thank my advisors, Mark Nidre and Dana Brooks, and the other members of the Biomedical Optics Research Group. Actually, Ying is the one that's building this instrument, so if you have any questions, I'm sure she'd love to talk to you about it. And then finally, I want to acknowledge our funding source. Thank you.
Okay, so I have a naive question. When you first posed the problem on your very first slide, the first thing that came to my mind was it's a source separation problem. So, and there's been a classical speech source or whatever. So can you just kind of broadly comment on what distinguishes your problem from those? I think it's more structured, maybe, but why would you not just throw sort of classical source separation techniques at this?
Yeah, so I mean, so people have done that in the field. They've tried to do blind source separation, but again, since we have an idea of what the signatures will look like, we want to rely on that structure. The people that have tried to do that with non-negative matrix factorization haven't been particularly successful. I don't think independent component analysis would apply. So yeah, there are a variety of other approaches. We really wanted to make use of the fact that, you know, we know essentially what the signatures should look like. There are just certain distortions that are not easy to predict. So
It totally makes sense.
Was there any other approach from a practical point of view to solve the same problem and avoid, I mean, the open literature? I mean, is there any other way than coming to these mathematical solutions for, you know, I mean, from a practical point of view to detect this, or?
You mean less mathematical than this?
No, just avoiding altogether. So this is non-invasive approach, right?
yeah.
Yeah, the measurement, it's not invasive except for getting the fluorophore in there.
Yeah. No, I'm just trying to find out whether one can have other approaches to solve the same problem from a
Yeah,
practical.
you could cut the mouse open.
No.
But I mean, I'm not even a joke, that's how most research is
Really?
done, right?
Yeah.
Right, most research, you
I
go to a
meant
pharmaceutical company,
non-invasive.
they sacrifice the animal.
Right.
The whole promise of this is to be able to do this in a way that you don't have to do that.
Mm.
So I'm not sure,
Yeah, I thought
I
without like some
I mean,
tomographic.
There are other imaging modalities.
My
So there's
imaging.
X-ray CT, right?
Right.
There's
Yeah,
PET
that's what I meant.
scans, there's, but I mean.
That's what I meant.
So
you know,
the
like-
advantage, so I didn't really have time to get into this, but
Yes.
one reason that people are interested in doing it with near-infrared light is that near-infrared light is non-ionizing. So you
Uh-huh.
don't have the bad side effects of X-ray.
I see.
Also, in terms of instrumentation, it's a lot simpler, the instrumentation is a lot simpler and less expensive than, say, MRI or
Mhm.
PET. So there are advantages those are the advantages why people continue to work with near-infrared light, despite the fact
Mhm.
that it's, you know, diffusive and the problem is still posed.
Yeah,
There's a
yeah.
third big advantage from a biological standpoint, which is the variety of contrasts you can see, so that you can target a huge range of types of molecules or
Mhm.
types of genes that get expressed, and X-ray and even MR are much more limited in terms of the
Mhm.
varieties of contrast you can get. So
Yeah.
biologists really like
No,
optical
she
imaging
answered
for
my
that
question.
Yeah.
That's good.
You talked about in your extended library too, having like several hundred signatures for each fluorophore. Do you ever see an overlap between different fluorophores or things like that? Or do they all have hundreds of unique signatures?
Well, I mean, so there's certainly overlap spectrally
Mm-hmm.
and temporally, but when you combine the two together, no, there's no, I mean, they can be very close. And I mean, that's kind of an open question. How close in lifetime would the fluorophores be before this fails? Or how close would the emission spectra could they get before it fails? I mean, I used 2 fluorophores in there that were only separated the peak emissions by 20 nanometers, so that's really kind of close. And also the lifetime is only a couple of nanoseconds, so that's also pretty close. Could you push them closer? I don't know, but yeah, I mean, so when you're looking at the signal in terms of both time and wavelength, you don't run into that problem.
So your subspaces disjoint then?
No.
I thought when I asked you that, you told me no, so now I'm a little confused by your answer to Gwendolyn.
No, but I mean, but the thing is, I mean, are they technically disjoint, and which parts of those subspaces are you sampling though, right? So if my subspaces don't have to be disjoint, but if I never sample outside of a certain region, I might be safe. So I have to say that, I mean, answers to questions like that, I mean, that is really open, because our problem is a little bit different from the problem that has been looked at in the sparse subspace clustering literature. And so I think it would actually be really interesting to look at our problem from that perspective.
I apologize because I was late, so I might have missed it. How is that you're using the temporal information that you have here?
So, I mean, I'm basically taking both the spectral and temporal information and vectorizing it. So I have a signature that's composed of temporal information for different channels.
If you shuffle your temporal information, you're basically using the same data, and you probably will get the same results.
I think temporal is decay.
There's a temporal, there's a temporal structure, so I'm not sure I know what you mean.
What I
sampling in
mean
time.
is that the relationship, the memory of the system, you know where I'm coming from probably.
No, I'm sure.
Okay, so what I'm saying is that if I look at just a trajectory, okay, as my information, it's different than if I look at one piece of the trajectory and what has come before. I'm looking at the past, at the sliding window.
Yeah,
So
but here you're taking an entire measurement
yes,
in time
yes,
at multiple wavelengths and treating that like a single measurement,
right.
right?
So what I'm saying, if you take that vector
Yeah.
and you flip the order, like if you do PCA on that data, if you flip the order of the frames and you do PCA on the same data, you basically will get the same answer.
But you would get, it wouldn't correspond to the same measurement, so I'm not sure
No.
what you mean.
No, you flip consistently, like every time, in every measurement, I flip the measurement at time 3 with the measurement at time 4. I create a new matrix. And I did a PCA of that. You get the same result. The subspaces you get is the same. What I'm saying,
Yeah, so if you flip, so if you reshuffle, if you reorder time consistently across the problem,
yes, that,
it wouldn't
it wouldn't make any
difference,
difference.
right?
So my claim is that with that, you are not exploiting to the full extent that you have your temporal data.
So-
So like if I did, and
So-
this matrix will be huge, so it's impossible to think about it, but if I did a Hankel matrix of that, then I would be considering the actual dynamics of how that data is changing.
So you reduce
Yeah,
the
although
problem to
it's not
dynamic
linear
situation.
Yeah.
decay, it's a nonlinear decay, so I'm not sure you can do that over. So there is an approach, as best I understand it, what Anand does, Kumar, who tries to fit the exponentials explicitly. So he uses a model for the temporal behavior, which is just as a decaying exponential, and fits the exponential coefficients and then decomposes things that way, right? I mean, I think that's.
But it doesn't have to be a linear, I mean, you can model that as the output of a linear system, right, that's decaying. So it doesn't have to be a
Mhm.
linear relationship.
I guess I'm not clear on why you say that I'm not exploiting fully the time dimension, though. I mean, what difference does reorder make?
So you're not modeling, I mean, I think I understand what you're saying, so I think what comes is,
Uh-huh.
you don't have a low dimensional dynamic model for what's happening in time. And there is temporal structure.
There's temporary, like the difference between measurement at t and t plus 1 may be constant. Let's assume that it's not, but let's assume that's a model, right?
Well,
And if that was there, your method is not exploiting that.
this is my model for what happens in time.
Right.
It's a mono-exponential decay.
But you don't actually use that.
But you're
model.
not using that in your,
Actually,
I am.
Yeah, she is actually, that's
I
right.
am, because I'm making fluorophore signatures that incorporate this mono-exponential decay. Those are the contents of my library.
So synthesizing the pattern which she's looking for, she's using that.
Yeah, I had to think about it.
Yeah, I do too. It's an interesting question.
I have a
You're
much
right.
easier question. So the mouse is small, right?
Yes.
So you said, 25 millimeters and you can get something around their torso. So how well does this, I mean, you try to do a person, they're a lot bigger than
Yeah.
25 millimeters.
So-
Is this something that easily scales up in dimension? I mean, obviously more easily than
Yeah.
trying to invert the matrix, but
Well,
what are, or just, and forget about your method, I mean, what are the big hangups in going
Yeah.
to people or larger things than?
so the big hangup is how much tissue you have to go through, right? So the signal is going to succumb to absorption and scattering at some point.
So if you hold the laser
people
pointer
can
up to your
do-
finger, right, and then hold it up, right, hold it up to your forearm,
Well,
you don't see as much light, you don't get enough light flow.
Right, right.
so let me just, it's not used in humans to image a torso, but it is used in breast, for example, diffuse optical imaging. So you can go maybe 6-8 centimeters, not really scaling up much more than that without doing something clever that's not being done yet, so.
And breast is relatively homogeneous, and is that a feature of it, as opposed to, say, going around your arm where you've got muscle and fat?
Yeah, well,
and bone?
the people that work on breast don't think breast is.
There's no bone.
Yeah, no, so the other main application for non-fluorescent imaging is brain function. So you can see well enough through the skull to see something about the
Yeah.
cortex.
Okay.
So that's pretty.
But again, like, you
heterogeneous.
couldn't go like from
But
one
yeah,
end
but
to
you
the
can't reach
other
very far,
in
exactly.
you can do it in babies, but not in,
Right,
so
yeah.
people do it on the same side.
Babies also because the skull is thinner, so your neonates are good.
Right, okay.
Yes?
When you say a sparse, you mean what, the vector A is a sparse, right?
Yes. Yeah, so actually, let me, because I didn't have time to go into this, but this is a really good question. So yes, what I'm saying is that the vector, I'm asking for a vector A that's sparse. But now,
So
that
is-
doesn't mean
Is
that
it
the,
sparse in a continuous space, in continuous parameter space, or it's discretized?
no, the discretized. So only a few components are non-zero.
I'm
He's asking.
physically, is your problem physically sparse, or you make it sparse and discretized?
So in constructing the library in the way that I do,
Yeah, you discretize
I
both
make it, yeah,
time and vector.
but what I'm doing is constructing a situation where a sparse solution exists to my problem, you see. Because what I'm doing is, you're the algorithm, your job is to find a vector that matches the measured signal well, right? Well, I've sampled so well for you that with one or two vectors, you're gonna get the job done.
And
So I create a situation with my matrix that allows for there to be a sparse solution to the problem.
if you increase the sampling frequency for both time and wavelengths,
Uh-huh.
so how much the resolution get improved?
Well, the resolution, in terms of demixing, the resolution is limited by the tomographic reconstruction, which is a whole separate problem. You have to regularize. What limits the resolution in the tomographic reconstruction is the physics of the light propagation. Instead of propagating in straight line, like X-rays, it's propagating in these volleys. So there's a certain point beyond which, due to the physics, you can't improve. So the limiting factor for the problem is not the demixing. we have the demixing errors now well below what we need it to be. It's the tomographic reconstruction.
Well, because there's a research about the super resolution effect. When you increase the sampling rate, therefore this resolution gets saturated at some point. So if you go behind that sampling frequency, you wouldn't gain
Yeah.
much.
Yeah,
So
But-
if you increase the library, I mean the dictionary,
right,
so after some point,
right.
there's no,
I mean, we're already down to 1 or 2% errors. Anything less than 10 will give us great tomographic reconstruction, so there's no point in increasing the resolution of the library. We already, I mean, you could even argue that we're already oversampled in a way,
yeah.
that I could get rid of some points in my library and still get good enough errors to do good enough reconstructions tomographically.
So what's the sampling frequency for your dictionary?
Well, so I do so I generate a signature for every fluorophore, for every source detector measurement at every pixel, okay? And then I prune signatures that have a total path length greater than some point, because that corresponds to places, locations where the fluorophore would simply be too far away from the source or the detector to generate an impact on that measurement. And then I prune signatures that are, I plot it in terms of distance to the source and distance to the detector, and I say I noticed through simulations that if I said I'll get rid of signatures that are closer together than, let's say, 5.5 millimeters, I can still get really good reconstructions. If I change that parameter, my reconstruction error, if I do coarser, my reconstruction error would probably go up, but you know, I'm being really conservative right now in the way that I'm doing it, because I'm getting 1 to 2% errors, and I don't really need such great performance from the demixing algorithm.
And you use OMP in your simulation?
No, I use Lawson and Hansen non-negative least squares. Or non-negative,
OMP.
which is a version of you can consider that a version of non-negative OMP. This is my claim. Well, one other person has claimed it actually in a paper published last year in Signal Processing Letters. There was somebody that said, oh look, there's this kind of similarity between these two, but he didn't do anything with it, he just...
Thank you.
Thanks very much.
She's comparing the vectors rather than comparing them the line dynamic with the vector.
So her argument back.
But depends on the initial position. When you generate, like if you have a different initial position, you may have a different